Let's imagine that the skin has been penetrated by a sharp object, bringing with it some bacteria that we see in blue. The bacteria has now penetrated the epidermis of the skin and it is in the dermis. The injured tissue produces alarm chemicals shown in yellow, and these alarm chemicals stimulate a cell called a mast cell that is shown in blue. The mast cell is a cousin of the basophil, and we know that basophil releases histamine, and histamines make capillaries leaky. So that's what we see here. The histamine from the mast cell makes the capillaries leaky so that neutrophils and macrophages can squeeze through the capillary and arrive at the site of injury. So that's what we see here, the neutrophils and macrophages arriving at the site of injury. So let's go over the steps one more time. The first step is that the tissue is injured by a sharp object and bacteria enters the skin and reaches the dermis. The injured tissue now produces a vast variety of chemicals, one of which stimulates the mast cell. So alarm chemicals are released by the injured tissue. The third step is that mast cells are stimulated. Mast cells are cousins of basophil. They are like the basophil and they release histamine. And histamine is known to make capillaries leaky so that more things can come out of the capillary. Once the capillaries are made leaky, the white blood cells can now squeeze out of them. That's step 4. The neutrophils and the macrophages are attracted to the site of injury, and so they move to that area and then they squeeze out of the capillaries and arrive at the site of injury. Step 5 is that the neutrophils and macrophages kill the bacteria via phagocytosis. A natural killer cell is a special type of lymphocyte that has the ability to carry out immunological surveillance. These cells travel through the bloodstream looking for cancer cells and virus-infected cells. Unhealthy cells such as these look quite different from the normal cells. These enemy cells carry special receptors on their cell surface that can be recognized by the natural killer cell. So let's take a look at how a natural killer cell destroys the enemy cell. The natural killer cell is shown here in pink and the enemy cell is shown in blue. The enemy cell could either be a cancer cell or a virus-infected cell. So when the natural killer cell comes in contact with the enemy cell, it releases proteins known as perforins. Perforate means to make a hole, and perforin is a protein that makes a hole. So that's exactly what it does. These proteins reach the enemy cell and make a hole in them. The hole is then used by the NK cell to introduce enzymes. These enzymes are small and granular, and so they are called granzymes. These granzymes make the enemy cell undergo apoptosis, meaning it causes the enemy cell to commit suicide. Once the enemy cell is destroyed, it is then cleaned up by a macrophage which carries out phagocytosis and clears the debris or remains of the destroyed enemy cell. Now let's see how a phagocyte destroys a pathogen and presents its antigens to more sophisticated cells of the immune system. So here is the pathogen in yellow and its associated antigens. You want to imagine antigens as if it were buttons on the coat of the pathogen. Here is the macrophage attempting to engulf the pathogen. We call this process endocytosis, or more specifically phagocytosis, because the cell is actually eating the pathogen. Once the pathogen is inside the cell, it is put into the lysosome. The lysosome then destroys the pathogen while keeping its antigens intact. These antigens, imagine they are buttons, they are worn by the phagocyte to say, I've just killed something and here are the buttons to show for it. These buttons are the antigens, and these antigens are presented to T lymphocytes, which are the most sophisticated type of white blood cell in the third line of defense. So the antigen presenting cell, or APC, is nothing but a phagocyte. Let's take a look at the protective protein called interferon. Interferon interferes with viral replication. In order to understand how interferon functions, we need to understand how viruses operate. A virus cannot multiply by itself. It must enter your cell and then use the resources in your cell in order to multiply. Once the virus multiplies, it then lyses the cell, the host cell, basically destroys it, and all the viruses infect the neighboring cells. This is how they operate. The interferon stops this from happening. So let's see how. We have two cells here. The cell on the left is being infected by a virus. The virus is shown in yellow. The virus does not enter the cell, but it introduces its genetic material. The genetic material could be either DNA or RNA. Once the genetic material enters, the cell responds by becoming activated and making a protein called interferon. Interferon is shown in pink. Once the interferon is made, it can now leave the cell via exocytosis and enter the neighboring cells. So interferon leaves the cell, reaches neighboring cells, and influences the neighboring cells and asks them to prepare for viral infection. So here's your uninfected cell. This cell does not know that there is a virus in the vicinity. Once the interferon signals this cell, the uninfected cell makes antiviral proteins called AVP. These antiviral proteins are like little weapons that are ready for a viral invasion. When the virus actually arrives and introduces its DNA or RNA, AVP blocks viral replication. If viruses cannot multiply, they cannot survive. So the AVP blocks the viral replication and basically stops viral infection. So let's recap. Interferon is a protein that is produced by an infected cell, and its job is to signal healthy cells in the neighborhood to prepare for viral invasion.